It's my pleasure to introduce Jeremy Owens, who's coming to us virtually from Florida State in Tallahassee, where it's definitely warmer than it is in Chicago this morning. Jeremy was a PhD student with Tim Lyons at UC Riverside, where he used a variety of geochemical redox proxies, which he can describe a little bit, I assume, to better constrain iron and sulfur cycling in the proto-North Atlantic, including during some of our department's favorite OAEs. And then he moved on to do a postdoc with Suni Nielsen at Woods Hole, where he expanded that analytical repertoire to include geochemistry and isotopes of a number of trace metal systems, and has expanded upon that work at Florida State, which we'll hear about a little bit more right now. So with that.
Awesome. Thanks, Andy. I don't know if you guys normally ask questions during it, but if you want to, that's fine. I'm open for that. Either way, or you can hold them at the end. So it's a great pleasure. I thank both Andy and Maggie for the invitation, and Northwestern, and everybody for attending. I am bummed I'm not in person. I'm sure we're all tired of Zoom meetings at this point, but so goes life. But I'm excited to talk to everybody and give some of our latest research in my lab group. So initially, I was really wanting to go big and talk about multiple metal isotope systems, and as Maggie said, elements that I've never heard of. I don't know if I've heard of them either. But as I started to make the talk, I said, oh my God, I'm never going to get through any of this. So I'm going to focus today on thallium isotopes, but we are working on others, and I'll talk a little bit about just the basics of vanadium isotopes, just kind of where it fits in. But if anybody wants to talk or has questions about that, I'm happy to chat about that more as well. And so today I'm going to talk about how these how thallium isotopes are, how we're utilizing this in the geologic record. And this work is really all of the team. I haven't done any of the lab work, I think, on any of this at this point, but really it's going to try to take what we've learned from all the different events. So it'll be kind of a snapshot, very quick snapshot on each of these events, kind of talk about the main highlights, and hopefully compare and contrast some of what we're maybe learning, or at least some observations that we're trying to unpack and tease out. And so I'd like to thank my postdocs that I've had since I've been at FSU, a group of grad students that have either been mine or worked with Seth Young, and a whole group of collaborators as well. So this picture is from I don't think I'll show it again, it's from one of the sections that we've been working on in Alaska, the Triassic-Jurassic mass extinction, which is, I think you guys can see my mouse. We actually camp up on this ledge, and the section is way down here, which we have to hike to every day and carry the rocks back from. And we obviously haven't been able to do that in the last couple of years with COVID, but hopefully we'll get back, considering it's an NSF grant, but at some point, I think they might end it and we won't be able to get back. All right, so I'll spend a very small amount of time on the TJ because it's very preliminary at this point. But, all right, there we go. All right, so just a brief intro about deoxygenation, or oxygen, and why we should care. As many know, this is obviously an important aspect is understanding oxygen, and certainly eukaryotes have a high need for it. But we also are starting to develop and get more data about current climate change. And so in 2018, there's been numerous different versions of this kind of figure, but it's starting to show that the modern oxygen, or the modern ocean, is becoming deoxygenated. And these are for a variety of reasons, including circulation, upwelling, but also nutrient inputs, and also warming, right? As the Earth is warming, and the ocean's warming, there's less gas solubility, so there's less oxygen that the ocean can hold. And so this may be of interest moving forward as another sort of cascade of events that might happen from a warming planet. But it's hard to look at this on our timescale. So a lot of what I do is really interested in how is deoxygenation played out in the geologic record. And so that's kind of what I'm going to focus on today, is really looking at this deoxygenation. And maybe it'll provide some aspects of what might happen in the future. And I'll give away the punchline. I hope that's not true for all of us, but we'll go through this and see what we've seen in the geologic record from major finding events. And so we know it has massive biological effects for this deoxygenation, but there's also responses that many organisms can have. So the figure here, B and D, is obviously catastrophic. So you can see fish kills here from a fishery in the Philippines. And in D, the bottom left, you can see coral reefs that have undergone bleaching during this process. So it's not just ocean acidification that causes coral bleaching, but also the loss of oxygen and obviously the organisms and the megareans being killed off. But there are organisms that have interesting responses to low oxygen. We've seen this in modern oxygen minimum zones, for instance. So you can see an organism here, the red being elevated iron, so they can actually deal with low oxygen environments much better. So they have a response to this. And some of these really small tiny organisms will actually move into low oxygen environments to avoid predation. So in some ways, you can see some organisms will do better under low oxygen at times, and we've seen this in the geologic record as well. But if it gets too bad, they usually kick the bucket as well. So, you know, there's a real interest in trying to understand this and what our future could hold from this record. So those that are work on OIUs will recognize this figure. It's relatively famous at this point, with Hugh Jenkins sort of putting together a group of feedbacks that may happen under one of these climate events. And I think it's pretty applicable to think about in terms of potentially our future. But a warming planet can have kind of a cascade of events of different feedbacks that has an impact that may, or most of them, amplifying the system and making it worse. But it also increases the hydrologic cycle, which then can increase the nutrient delivery to oceans, eventually driving increased primary productivity, consuming available oxygen. And so therefore having some loss of oxygen during this process. Eventually, if there's enough loss of oxygen, you might preserve and bury more organic carbon. And that sort of is a positive feedback to sustain anoxia, or eventually euxinia, which is sulfidic water columns. And I would say this is probably a relatively simplistic view of this. There's lots of other feedbacks that are going on. It's not quite a one-way street or linear, but it's sort of the idea and the concept in terms of understanding how oxygen plays a role in this, and what the feedbacks might be as well. And obviously, you have to have something to start this off or kick this off, which is always debated for all the events. They're probably different for each one, but there's certainly some commonalities looking at events where you bury lots of organic carbon and what the redox response might look like. And so if you look at the modern ocean, while any of us would say it's an oxygenated world and atmosphere, and that's dominantly true. It is mostly oxic, most of the ocean is oxygenated. There are complexities and variability. It's not a homogeneous world. And so there are areas where we have low oxygen environments. And so famously, these are sort of in upwelling regimes, both at equatorial and on the western edges of continents, in the northern hemisphere, for instance, and in the southern as well. So these are areas where you have very low oxygen, usually just below the photic zone, where you start seeing this oxygen minimum zone, or OMZ. And so this is happening naturally on our planet, but you can imagine that a deoxygenated ocean, or times where you start losing oxygen, that this might become more expanded and also in more reducing. So not just that you have low oxygen, but you might even get to areas where it becomes more sulfidic or euxinic in them as well. So this is kind of the concept that you sort of amplify the system that we normally have in these events potentially. And if you look at the amount of organic carbon that's buried in the modern ocean, you can see that this map looks very similar to the previous OMZ. So the areas where you have high oxygen, or lots of oxygen minimum zones, more widespread, you bury a lot more TOC on the surface sediments as well. And so these are very closely linked between them. So the concept or the idea is that when you bury more TOCs, it's likely more reducing as well. But that's probably a bit simplistic. There's lots of areas for more complication than just a direct relationship between TOC and oxygen. And so I'm going to talk, like I said, I'm going to talk about a number of different events today, kind of give a snapshot of this. And so these are the 7 different time slices I'm going to look at. And so I'm going to really start with the mass extinctions that we've looked at. So there's 5 mass extinctions. We've analyzed work on 3 of them now. So the late Ordovician mass extinction, the Loam event, the Permar-Triassic, and the TJ boundary work that we've done. So these are highlighted in red. There's big changes in the carbon isotopes. So the carbon cycle is variable at this point and dynamic. And I'm going to compare these to 4 smaller, I'm going to call them climate events today, sort of usually associated with some sort of extinction or biological turnover, but not nearly the size of a mass extinction. There's also significant carbon cycle changes within them. And so I'll talk first about OAE2, everybody's favorite, Northwestern, right? And then another OAE, the Torresina OAE. And then a couple in the Phanerozoic, the Lau event and the SPICE. I'll spend very briefly on the SPICE, the Steptonian positive isotope carbon excursion. So it's a mouthful. All right, so before we get there, let's talk a little bit about the redox ladder. So this is really key to a lot of the work that I'm going to talk about. And so this is just the classic redox ladder from Froelich in the 80s, and I should say late 70s. And so this is just a generic representation of when you lose oxygen, organisms, especially facultative anaerobes, will switch to other electron acceptors. And so you can get to using sort of nitrogen and have nitrogen reduction. You can then have organisms also use manganese. So these are manganese oxides, and they release dissolved manganese. We're going to spend a lot of time talking about manganese for the rest of the talk. So the reason that I am interested in it is very high on the redox ladder. So it's very close to where oxygen reduction occurs. So once you are running out of oxygen, many organisms will switch to manganese. You can also look at iron and sulfate reduction. And all of these are really important in different aspects. Sulfate reduction is a little farther down the redox ladder and certainly important, but not at the earliest response to changes in oxygen. And so I like to think about what this means in a burner classification, where he sort of used this as you have oxic conditions up at the top, and then all of this, everything below that is anoxic. So you can split the anoxic zone into different he has them in 3 different zones, where he considers it post-oxic. And so that's anything that's in this manganese, nitrate, and iron reduction zone. And then eventually you go to sulfidic conditions, so you'd have hydrogen sulfide. Below that is the methanic zone, which I've taken off this, so I'm not going to spend discussing anything about that part of the redox ladder. And so you can map this out and think about what this means. So today we have anoxic portions of our ocean, those OMZs, but we also have areas that are sulfidic as well in the water column, and those are the 2 biggest basins, being the Ariake Basin and the Black Sea. And so you can split those into 2 different groups. And so you can kind of think of this as anoxic is one sort of generic term. And you can kind of break this up into post-oxic and sulfidic, and then here's methanic. But a lot of people will just say it's anoxic, which is fine. But we can probably start parsing our information a little bit more finely than just the generic term of anoxic. And I'm guilty of this as well. I have plenty of publications that have done this. But what I'm trying to do moving forward is be a little bit more, breaking these up and being more specific on this. And so what we can do is we have a whole host of geochemical tools at this point, which most of the time overwhelms all of us. I'm trying to think about it. But I'm just going to do a couple of things just to highlight is some of these tools are really great at telling us about local redox conditions. And that's required. We really need that. It's not that none of these are better or worse than the other. They just tell us something different. So things like rare earths or iodine, calcium, manganese concentrations, all of these tell us something local, but they also tell us a specific part of the redox ladder. So there's not a one size fits all, but they can give us different information about where we're at in the redox ladder. And then you have some that are, depending on the system, can be used for local or global. Combination of proxies might tell us some information about these as well. And so these are quite useful. I should point out that the black is for organic rich shales typically, and the gray bar is typically found in using carbonate lithologies. And then on the right is the proxies that we're trying to use to tell us about global conditions. And so many of those people in the department have used, so carbonate associated sulfates, sulfur isotopes, uranium isotopes, molybdenum isotopes, all quite useful. And then today I'm going to talk mainly about, or completely about thallium isotopes. And the reason, again, is because thallium isotope is up on the redox ladder. So it's one of the first responses to low oxygen conditions, because it's really tracking, as I'll tell you in a bit, manganese reduction. And so it sort of falls in an interesting part of the redox ladder. And it's telling us about the global manganese oxide burial, which relates to the oxygen. A couple, I'm not going to show because I don't have time, but all of the sites that we use, we have to know the local redox conditions. Thallium isotopes does not help us with that. So they're all paired with some other portion of information to tell us about the local redox. We need to make sure that there's no manganese oxides in those samples, because that would completely throw off our interpretation. But so we use things like iron speciation and manganese concentrations within our samples to make sure that there's very low manganese oxides that and that we wouldn't be analyzing that. So just keep that in mind. It's not, we're not just taking a sample and just doing thallium isotopes. There's a whole bunch of work behind it, but I'm not going to have time to discuss all of that today. And the bit that I wanted to talk about, which I don't have time as well, is vanadium isotopes. And so again, it's high on the redox ladder. It looks like it's probably telling us, probably the latest data is probably moving over into the local, telling us about local conditions, but some really interesting information that we've started to look on that as well. So if you're interested, I can talk about it at the end or in one-on-one. All right, so talk a little bit about the mass balance. This is a really new proxy, and nobody really knows anything about it at this stage. And we're still learning a lot. So I would say, we're still working on the modern mass balance. It's pretty well put together, but there's still, as always, more to learn. So you'll see that all of our work will be in delta, or sorry, not delta notation, but epsilon notation. So epsilon notation, and the only difference of that, delta notation or any other isotope system is it's just the difference of 10,000. So it's a really small difference that we're measuring, but we can do it with really high precision on our instrumentation. And so this is what we'll look at. All right, so the seawater, mainly I'm going to focus on is the isotope values, not the concentrations. But the seawater value of the modern ocean is -6. And so that's because we'll talk about it's a little bit, it's well oxygenated, so we get values that are -6. Importantly, one of the really big keys of thallium is that the residence time is relatively short. It's only 18,500 years, so roughly around 20,000 years is the residence time. And so that allows us to probably have some of the earliest perturbations recorded, because it doesn't have a long residence time. But importantly, it's also longer than ocean mixing. So if you look at modern ocean and you do in the oxic ocean, you look at dissolved manganese, or sorry, dissolved thallium and the isotopes, it's homogeneous throughout anywhere in the oxic ocean. So that we know of. Sure, there'll be an exception at some point. All right, so let's look at the sinks, or sorry, the sources first. And so there's been a lot of work on various different sources. So they've sort of been grouped together. So things like volcanic gases, has a value of about -2. That -2 you'll see is common thread. It's also the continental crust value, and it represents about 37% of our current source input. Things like mineral aerosols from, so things from dust, also, because the continental crust is about -2, has a value very similar, relatively small source. Rivers, again, value of -2.5, about a quarter of all of it coming in. Hydrothermal fluids, again, -2, and a source of about 17%. And so those four are values that are very close together. So that means that any changes in the isotope value we are observing likely aren't coming from changes within these four systems because they have no isotopic leverage. Makes it useful because we know almost all these climate events probably have changes in their inputs as well. The last one is pore waters. And so these are where basically you have reducing conditions in like an oxygen minimum zone, where you might release some of those manganese oxides and things back to the water. You get a value roughly at 0, which is about 17% of the source. This value is not well constrained. We have a couple of projects working on this, and this, based on that work, it's fairly close. So it's not going to change the stories that I'm going to tell today or talk about. Importantly, the sinks that we have, there's two main sinks. There's a third one as well, but it's really small. So there's two main sinks: altoceanic crust here on the left, which has a value of -12 to -6. So that's of the actual epsilon values. If you compare that to the seawater, that's where that number is coming from. So the difference between seawater and that value, the average is about -1.2. So it's a really small fractionation, but it's a big sink. It's 63%. What we should be sort of recognize here is important is that this is essentially the subducting slab. And so to make big changes on short time intervals, especially if they go in multi-direction or go back and forth, it'd be really difficult to do on short time scales. But it certainly probably has a pretty big role on longer time scales if you're thinking of the entire Phanerozoic, for instance. But for short-term climate events, what we mainly have focused on, because you can change them quite rapidly, are manganese oxides. And so manganese oxides have a large fractionation from seawater, or I should start with the actual isotope values from +6 to +12 today. And from seawater, the average is about 16, so +16 from the starting seawater value. And it's about a third of the entire sink. So the other couple of sinks that are relatively small is this euxinic sink. So the euxinic sink, really importantly, this is essentially what we're going to try to use for our black shales. It captures the seawater value. And so that seawater value we can use in the ancient record to reconstruct what the ocean looked like and be able to make some hypothesis to what the sources of that change would be. And so it's a small sink today and no fractionation. The other one is the suboxic anoxic version over here. Probably represents not very well constrained, maybe 1%. And there's a variable isotope range from anywhere from 0 to +6, and that's likely because these are areas that are incorporating both seawater values and some of that manganese oxide, because it's not entirely reducing. But it's a relatively small sink that we understand today. So this is the mass balance that we'll kind of work from to give an understanding of these kind of events. And the last part of this is looking at where manganese is buried today. And so these are sort of the deep abyssal plains where most of this manganese is buried. But I really want to point out that when you see this, these are the large nodules, right? When you get these big pieces of manganese oxides. And so these are the deepest portion of the ocean where you have very low sedimentation rates. But that doesn't mean that there's not manganese oxides being buried along the continental margins. They're just being buried with lots of sediment. And so there's not these nodules that are occurring as well. So it's not incredibly well constrained on terms of the mass balance of manganese. And the reason is because manganese is inherently local. So you have to drill everywhere to get a good idea of manganese concentrations in sediments. And there's no individual isotopes that are long-lived for manganese. So it has one long-lived isotope, And so that's why we're using thallium to sort of track the manganese as a way to do this. So it's, if you want to be cynical about it, we're using a proxy to track the proxy, but get at oxygen. But it's our best way at the moment. And so hopefully we can learn something from these methods. All right, so I'm going to jump into these different extinction events and kind of walk through. It'll be relatively fast in terms of what we're going to talk about, but I'm just going to kind of highlight a few things, certainly ask questions as we go. So this first one I'm going to talk about is the Permo-Triassic mass extinction, or at the boundary. We've measured 3 different sections. One of the things we try to do as much as possible is measure in different places to make sure that we get a similar stratigraphic and magnitude shifts in the thallium. And they're all measured from the Panthalassa, which was the most significant ocean at that point. And unfortunately, we don't have anything from these other ocean basins, but I think at this point, you know, it's sort of a start, and we'll continue to refine this. So the Permo-Triassic being the largest mass extinction in Earth's history was a kind of a basic place to start looking at this. I should also point out that I'm not going to show all the other people's work that have done redox proxies on this. Recognize that we're not the first to say it's reducing. All of these events have had many other redox proxies, and there's we've known for quite a while using different systems that this is a relatively reducing time interval. But what I'm going to show is a little bit of variation on that and what we're understanding. So just recognize I'm not saying that nobody else has done a lot of other work as well. But using the thallium isotopes, we've looked at these 3 different sections. There's based on sedimentology and a few other age constraints. There's some differences in terms of what we can analyze, but I'm going to just highlight one of these. So just recognize that our interpretation is based on 3 different sections as much as we can. So Opal Creek is what we looked at here. There's a bit of a hiatus beforehand, and so many of the other sections tell us a little bit more about the earlier portion, at least being a continuous stratigraphic record. But what we were really interested in is that when these values become closer to -2, it suggests that it's more reducing because you don't have manganese oxides that are very positive, changing the seawater value. Interestingly, the surprise for us was the EPME, which is the in-Permian mass extinction. So this is where has been refined where the mass extinction really occurs, not actually at the boundary of the Permo-Triassic. You get really close to this, depending on how age dates you work. You get this kickback of the thallium isotopes that get back to values that are very close to modern, -6. And so this suggests that the only way to really do this over a really short time interval is to be bearing a lot of manganese oxides. And so you could say, well, you could bury a lot of manganese oxides if you just had a lot of manganese in the ocean, which is true, but manganese has to have oxygen all the way to the sediments. And so this suggests maybe not the whole ocean, but certainly some portions of it was allowed to bury a significant amount of manganese oxides, suggesting that it became, at least at some level, more oxygenated at the in-Permian mass extinction. And this is a paper by my PhD student that was just published last year in Nature Geoscience. And you can imagine that this was discussed and debated quite a lot, but there's really no other way to drive this value of -6 at the boundary. And so what this suggests is that it was probably relatively reducing well before and the extinction started, but the in-mass extinction might actually be not just anoxic, but it might be more heterogeneous and more variable than what we currently would have predicted. And so it may not just be that you have to have anoxic conditions, but maybe this is becoming more reducing as well. And so, or more oxic, followed by more reducing. And so after the in-Permian mass extinction, you see values that go back to the value kind of close to -2. And they stay there for quite a while. Actually, we haven't seen that they come back. It takes, maybe takes multi-million years for it to come back to these more oxic conditions. And so if you put all this together with some of the other data that's out there, interestingly, there's a whole bunch of wiggles in the carbon isotopes. It's
complicated because of volcanism. But interestingly, the oxygen isotopes actually suggest that it cools right at this time as well. So just a little blip, maybe a few tens of thousands of years. Uranium isotopes actually suggest starts becoming more and more reducing. And thallium here suggests that it's actually slightly more toxic during that time and bearing more manganese oxides. So how do we sort of reconcile all of this in a model? Well, this part is certainly the interpretations based on all the other data. But what we think is occurring is in the middle Permian, you have values that are close to modern seawater values, -6, so it's relatively well oxygenated. And we have some areas that are bearing pyrite, so we can get these values. But you can see that most of the manganese is being deposited in the sediments, very little dissolved manganese in the ocean, much like today. And maybe as you go into the latest Permian, evidence from volcanic proxies suggests that the Siberian traps are starting to release material, starting to develop. And so there's more CO2 in the atmosphere, it warms, you get more reducing conditions. And you can see seawater values of about -3. Temperatures are increasing. And now you have less of your manganese being deposited in sediments and more dissolved in the ocean. And that onset of the EPME here in the bottom left, you actually might have evidence for SO2 release, and SO2 is a good way to reflect some of that light and actually might have some cooling effect. And that cooling effect is suggested by the oxygen isotopes. So you can actually have more oxygen in the water, depresses that chemocline down a little bit. And so it becomes probably not as oxygenated as before because you have a lot of manganese floating around from before, just before. And so you bury some of that and suggest large manganese oxide burial. And then you go back to SO2. It's probably been taken out of the atmosphere and back into CO2, and you become more reducing again, and it's a warmer temperature. And so this part of the EPME that we're suggesting could be because of the materials that are coming out from volcanism. We don't have specific evidence for that, but others that have worked on this and modeled it have suggested this previously as well. And so we just sort of put our data and our observations into this context. All right, so that's all I'm going to talk on the Permo-Triassic. There's a lot to really pull out of that, but move on to a different mass extinction, a late Ordovician mass extinction. And again, we have multiple sections in this ocean, and this is about 440, 450 million years ago. It was a much different world at this point. But let's see what our thallium isotopes come up with. And you can see that, again, we can correlate to different sections and look at some of this. I'm going to just show one section from this. And this is some work that's in review by Nevin Kozik in Science Advances currently. And so what you'll see is something kind of similar, is that it's relatively reducing well before the event. And so the event here, the first pulse of it is this first dashed line. There's 2 pulses of the extinction, second dashed line. And so what you see is reducing conditions that become even worse right before this first extinction. But you see a return back to less reducing conditions, maybe something slightly more oxic. It stabilizes, and then again at the second pulse, you actually become more oxygenated than you have previously. And so this suggests that you have quite, again, a lot of variability of changes in oxygen right before or right during some of the pulses of mass extinction. And so this is an interesting observation. We didn't expect this to see it again, especially because the drivers are very different for the loam versus the Permo-Triassic. But it does suggest, and it's not as big of a change in this section. One of the other sections is the larger change, but it does suggest that there's more oxygenation in this. And so I think, for time's sake, I'm gonna skip through a little bit of the conceptual model, but it's very similar to the Permo-Triassic in terms of this changing of oxygen minimum zones, expanding and contracting with time, and how this will affect the biology and the extinction event potentially. I'm gonna come to that. And so what we'll see here in the TJ, this is a very preliminary data set. We're still continuing to work on it. A new PhD student is it's just started this work. But we're going to look here at Rangalia that was in the middle of the Panthelastic Ocean for this extinction event, has moved northward and it has collided with Alaska now. So this is our Alaska work that the picture was showing. And it represents an open ocean section that's the largest ocean at that point. So it's a lot better section than many others at this point in terms of the largest ocean basin. And so it's just, we only have one section from the TJ at this point. But you can see there's carbon isotope excursion that we just recently published. And that the extinction occurs somewhere in this green interval. We don't have perfect biostrat within the extinction interval, it's always difficult, but it occurs somewhere within this. And so interestingly, at the TJ, we have a really long section in Alaska, so we can go well below the interval, and you can see values that start off as relatively oxic, much like today, very similar values. And it starts moving, and admittedly, we need to fill in more gas here. But it starts to fill, this trend line starts moving towards very anoxic conditions, getting to values of -2. As you're approaching the interval of the mass extinction. And then as you actually head into the mass extinction, again, it actually heads back to very oxic conditions, bearing lots of manganese oxides, suggesting slightly more oxic conditions before heading back out to more anoxic and eventually maybe back. And so again, this is very preliminary. We have lots to do on this, but it does suggest that there's counterintuitively almost that there's an oxygenation during these mass extinctions. At least, it's at the upper portions of the redox ladder. So just a real quick sort of capture on the commonalities. So based on these five mass extinctions, thallium isotopes are highly dynamic and very variable throughout this. And so there's specific changes. And this is closely linked to manganese oxides, which we then interpret to changes in oxygen or deoxygenation. All three suggest that oxygen is depleted well before the event, but generally correlate with the initial diversity decrease, which I didn't talk about. But the extinction is an interval where most of the organisms go extinct, but there's certainly a longer term organism or record that organisms are going extinct before that as well. And importantly, within the prescribed extinction event, there's actually short-lived increased oxygenations that likely go to even more damaging reducing conditions. And this isn't happening. I'm not stating that oxygen and what we're showing is driving all the extinction. It's probably a combination of multiple stressors. And so paleoclimate, sea level, habitat, variability, and redox all are creating these turbulent conditions leading to the mass extinction. And so I want to compare that to what we're seeing in these shorter term climate events. And so I don't have to probably introduce much on OAE2, but here's a quick map, all the different sections and sites. And so some work that was published by, at the time an undergrad that was working with me as an REU student at this whole Chad Ostrander, published some work on this, and we looked at thallium isotopes in dim rare rise. And you see this big change. And it stays relatively constant. There's some wiggles, but it goes from about -4.5 to close to -2. And it stays relatively stable. In this record, we don't see it return back to baseline. We have some longer term records that suggest that it does eventually. And so this suggests more of a unidirectional change. And the other important part here is that it suggests a change before the carbon burial event occurs. So it's probably priming the system, or one of the leading edges to priming the system to help bury more organic carbon. And so what we've suggested on this is that there's a lip activity previous to this. There's an expansion of marine deoxygenation, eventually leading to anoxic sediments, and eventually you have OAE2 where you bury lots of organic carbon. And it's probably the culminating part of really this climate change. And I know Brad and Matt have published on some of this concept as well, is that there's probably a longer term record of carbon burial and during this event, and that we really just focus on kind of the main culminating event, probably a longer term record. But I should say is that there's no extinction that's associated with OAE2. There's a turnover event. And so maybe there's a difference between sort of what the thallium isotopes are suggesting at a mass extinction versus more of a significant climate event, but one that's maybe not as catastrophic biologically. We can also look at the Torsen OAE, which does have a smaller, but known mass extinction associated with it. And we looked at multiple sections here as well, but I'm just going to show you one. This one has some of the background information of iron telling us about local conditions. But importantly, here we have a long-term record. Again, values interestingly start near modern seawater values, leading into this carbon, sort of positive carbon isotope excursion that we first see. There's a reduction in oxygen and suggests that there's less manganese oxide burial. And then for the Torsian OAE, which is the part that buries lots of organic carbon, you see this negative carbon isotope excursion. You actually see where it kind of comes back to more oxide, but it's more stable, much more stable than what we might see in these mass extinctions. And it's more of a unidirectional change for this as well. And so this earlier portion does explain some of the extinction record that was happening previous, which people had noted, but really didn't have a driver for as well. So that might be a big cause of this. And you can see that here in this figure is that the species diversity on this top takes a hit before the mass the mass extinction, and then eventually, takes a hit before the Torsian or OAE proper, where the carbon isotopes are discussed. And then it continues to get worse during the event, likely because of increased or euxinia, for instance, before eventually coming out of it and having more oxygen conditions. And this is also fairly well constrained with volcanism. We have the initial volcanism actually occurring back here at the very start of this. And the main sort of event, I guess, of volcanism happened during the Torsian OAE itself. All right, so I've got just two more quick events that we'll talk about to make sure we have time for questions. But so going into the Paleozoic records, we have a few other time intervals there as well. And this is a slurring event called the Lau, which there is a significant mass, well, I shouldn't say mass, but there's a significant extinction, maybe the 7th largest extinction in the geologic record occurs during this event. It's been understudied in a lot of ways, even though it's such a large extinction. So we've worked on and published some work from this event as well. And so one of the things I want to point out first is you can look at the extinction interval, which, well, first let me back up. So you can first look at the carbon isotope excursion, much like the OAEs that you will know, and the CIE peak interval is shaded out by this gray interval. But what you might notice is that before this gray interval is that you have some of these organisms that are taking a hit or going extinct before the carbon isotope excursion. There's been long-standing debate if and why that occurs. And it wasn't something that we set out to do, but it looks like our data may help to explain this as well. And so it turns out here at this smaller event, it's reducing, certainly in comparison to today. There's less manganese oxide burial. But this gray bar represents when that extinction, the first initial extinction happens. And you can see a big state shift and thallium isotopes, suggesting more reducing conditions before the carbon isotope excursion. And then it stays more or less reducing throughout this condition. And you don't see this swing back to more oxygenated conditions like we would for the mass extinction. And so this may help explain this initial extinction event that you see, that you might have increased anoxia a little earlier than what we see from the carbon burial, so the positive carbon isotope excursion. And so this interval happens, and you can see that the carbon isotope interval more or less matches up with the sulfur isotopes. And so likely carbon and sulfur, as we know, have a fairly close relationship.
helping to drive the burial of lots of organic carbonate and pyrite. But anoxia or deoxygenation might occur a little earlier than those have suggested as well. So the last one I want to show, which is work that's in prep at the moment, is working on the SPICE event, which is right around 500 million years ago. And it's a very large carbon isotope excursion. There's also a number of trilobite biomoons or extinctions that are associated with this. And so interestingly for this event, you get values that are sort of in between, not quite modern oxic, but not anoxic. But just before the event, it becomes oxic and then goes into this sort of anoxic realm and kind of stays there and stays flat during this. And this suggests these very reducing conditions, but there is maybe a perturbation from kind of reducing before to oxic and then back. And so it's suggesting that it's highly variable. And we have two sections that show the same exact record as well. So different stratigraphic records, but, or differences, I should say. So what I'm kind of thinking at this point of what this might mean for these climate events is that not all, but many of them are associated with volcanism. And so you might have these sort of increased stepwise volcanism, and largely the main phase of that volcanism occurs with this gray bar, which is representing the OAE, the organic carbon burials, which we see by the positive, typically by positive carbon isotopes. Not always. Some of them, like the taurursine, can have a negative complication for a different day. But anoxic might actually, so this post-oxic conditions might actually increase early. So it's anoxic, but at this point post-oxic. And the euxinic conditions, part of that anoxia, occurs at the main phase of the carbon isotope excursion. You can see that by numerous different records, maybe trace metals or pyrites, or I should say CAS, carbonate associated sulfur, that's related to pyrite burial. And if you look at the extinction records in detail, for those that we have them, is that it seems that the high requirements organisms are taking a hit early. They're starting to actually decline much sooner than the actual carbon burial event. Eventually, coming back, organisms that have moderate requirements don't take much of a hit until sort of in the middle of these events. And interestingly, these low tolerant organisms actually do better, probably because there's less competition, but eventually, maybe because of rapid changes in oxygen, but also likely due to increased sulfidic conditions, they end up taking a pretty big hit as well, depending on which event you're talking about. This is obviously sort of generic in terms of all of the events. And so I kind of look at this as, if you take the modern ocean and think about how this might expand and contract with oxygen minimum zones, and where you might expect this, it's probably not necessarily the entire ocean, but it's also more dynamic, right? We put these pictures up here as 2D, but there's a time component. So they're probably expanding and contracting in different ways, and water mass is moving around. And so even for OAE2, where it suggests that it's very reducing, that might just mean that you don't bury a lot of manganese oxides because you have sediments that are reducing, or you might have a water mass that for some time is reducing enough to dissolve those manganese oxides. So it has a signature that's very low oxygen. It doesn't have to be that it's like that forever. It might be far more dynamic than that. So based on these four climate events, thallium isotopes are certainly less variable than compared to these extinction events, and there's certainly more of a unidirectional change. So it suggests that it's sort of maybe a one-way street, where if you want to have a mass extinction, or maybe you don't want to, but you have a mass extinction, it might be that there's far more changes in the redox environment. And this is catastrophic, but not to the level of a mass extinction. And all these events suggest reduced manganese oxides before and sometimes, many times after the carbon isotope excursion. So the precursor to more potent redox conditions during the event, such as sulfidic conditions. And this may help explain some of the extinction or turnover records that we've had previously that have been debated about potential causes or mechanisms. So I just have two quick slides. So thinking about this, what I became sort of wrapping my head around with all the group's work is kind of comparing and contrasting some of these events and the sizes or the magnitudes of these
different events. And so it's really started, I think, my take is on the currently with the observations that we have that the texture and the stratigraphic variability observed, both in the paleontological record and now some in the geochemical record start to make more sense. But there's also very different records between a mass extinction event and these smaller but significant climate and extinction events that we see. And so the severity and duration are important for these, certainly for mass extinctions, but also maybe tied to rapid variability. And it might not just be just a single oxic to anoxic conditions. There's certainly organisms that might evolve and do better under low oxygen conditions, and oxygen coming back into the system might be quite toxic or prevent them from doing well, and certainly help cause a mass extinction or be part of it for these long-term events. And so potentially the mode and the tempo of these drivers are important. That's sort of a big question, and everybody wants to understand is what the size and the type of volcanism probably is important. So things like, are they subaerial versus subaqueous? What they go through, how that might be different. Certainly, at least in the PT, it seemed interesting. But these mass extinctions, I don't think for any of them, and climate events, they're not all driven by the same sources or the same types or the same durations. So it's hard to put one, call them, you know, there's one thing that is causing these extinctions or is very important. So I just wanted to put up some discussion things that I'm sort of thinking about. This pairing redox proxies together will continue to provide a better constraints on the spatial and temporal records and give more of a holistic understanding of the redox picture. There's not, I've discussed about thallium today, what we're working on, but I don't think there's any one perfect geochemical tool, and it really needs to continue to knit together and utilizing the same samples, have a better picture of what the world might look like. And that includes the use of local and global redox. While I've talked about what I think is happening globally today, I can't tell you where this is happening at all at this point, right? And so you really have to build that local redox conditions to better understand where this might be occurring, because that's important. If it's a deep ocean, we don't have much life, right, in the deep oceans compared to what we have on the shelves and what's going on in those environments. And so can some of these differences be due to subaerial versus subaqueous? What kind of role does the type of volcanism have? And then the big question is always, what are the likely drivers? Probably are multiple stressors. There's not probably a one size fits all, but these understanding the magnitudes and the timing and these associated feedbacks are certainly things that will continue, and everybody, I think the community will continue to try to understand, but the links between things like weathering and how that might lead to nutrients and oxygen is an important aspect to better understand how these events sort of manifest themselves and what that might mean for our future. And so with that, I'd just like to say thank you guys for attending. I know it's yet another Zoom meeting, but I really appreciate having your attention and talking about my research. It's always fun to do. And then of course, some funding that I've had over the last 6 years. It's been great, and I look forward to hearing some of your comments and talk about science with you.